Okay, let's unpack a topic that, well, it sounds inherently dangerous, doesn't it? But it actually holds some incredible biological surprises.
Uh,
We're diving deep into the world of toxic animals today.
yeah, we're looking at everything from the ones you definitely shouldn't touch or eat to those that might bite or sting you.
And then kind of flipping it around to see how these exact same toxins are, believe it or not, changing medicine.
It's a fascinating area.
You hear people casually use terms like poisonous snake or maybe venomous frog
Right.
all the time.
Oh, constantly. And that misuse actually flags something really important.
Yeah, what's that?
Well, there's a crucial fundamental difference between venomous and poisonous. And understanding that difference, it unlocks a whole new way of looking at how animals use these toxins for survival.
Okay, so it's not just semantics. There's a real biological distinction.
Exactly. The core difference isn't really about how bad the toxin is per se. It's all about the delivery system. It's how that toxic substance gets from the animal into you.
Right, the mechanism. And there's this fantastic simple rule I read in the sources that really helps you remember it. How does that go again?
Ah, yes, it's the quickest way. It goes like this. If you bite it and you die, it's poison.
Yep.
But if it bites you and you die, that's venom.
If you bite it, poison. If it bites you, venom. Got it.
It tells you straight away whether the animal actively delivers the toxin, usually through a bite or sting that's venom, or if you have to passively receive it, maybe by touching or eating it, that's poison.
Perfect. So to explore this fascinating difference and, well, everything that comes with it, we've pulled together quite a stack of sources.
We have indeed.
We've got some foundational articles explaining that exact venom-poison distinction.
Then there are these comprehensive lists just showing the sheer variety of animals in these categories.
Yeah, some really surprising ones on there.
Definitely. And then there's a really detailed look at the intricate machinery animals use for delivering toxins. We'll focus quite a bit on snakes there.
Their systems are pretty incredible. And maybe the most surprising part, we've got sources diving deep into the cutting-edge research showing how these dangerous compounds are being turned into life-saving medicines.
So our mission in this deep dive really is to unpack these distinctions for you, look at some truly wild examples you might not expect, understand the clever biological tools involved, especially in delivery,
Right.
and then shift gears completely to appreciate the incredible potential these toxins hold for science and for health. It's kind of a journey from danger to discovery, you could say.
I like that. Danger to discovery.
Mm.
Okay, let's jump right into that core difference then, how these toxins actually get delivered. It really all comes down to the mechanism, doesn't it?
It absolutely does.
Mm.
Think of it as active versus passive transfer. Venomous animals are the ones with the, let's call it, biological injection system.
An injection system.
Yeah, they have to create a wound. Usually it's a bite, maybe a sting, or sometimes a sharp spine, or even a claw. They need a way to get their toxin into your body.
And the toxin itself, the chemical nature of it, sort of requires that injection.
Right. Precisely. Venom is typically made up of relatively large molecules, things like complex peptides and proteins.
Like a big molecule.
Big molecules. And these aren't great at just, soaking through your skin or the lining of your mouth. They need a direct pathway into your tissues or bloodstream to actually do their damage. So the animal needs a tool to push it past your body's outer defenses.
Makes sense. So what's the point for the animal? Why evolve such a complex, delivery system?
It's usually for one of two main reasons: getting dinner or avoiding becoming dinner.
Okay. Offense or defense?
Exactly. Many predators use venom to quickly paralyze or kill their prey. It makes the hunt much easier and, frankly, safer for them.
Less struggle.
Right. And for others, venom is a really potent defense mechanism. A painful bite or sting, that's a pretty effective way to tell a potential predator, maybe not.
Yeah, I can see that. So the classic examples we think of are things that bite or sting us.
Exactly that. Rattlesnakes delivering it with a bite, spiders using their fangs, bees and wasps with their stingers. Even marine creatures like the stonefish using sharp, venomous spines. They all have that specialized tool for active injection.
Okay, so that's venom, the active injector. Now let's flip that. Let's talk poison. This is the passive side.
Right. Poisonous animals, they don't need to wound you. Their toxins get into your system if you touch them and it gets absorbed through your skin, maybe if you inhale tiny particles, or most commonly if you try to eat them.
Ah, ingestion.
Yeah, ingestion. The toxins get into your digestive system. No injection needed at all.
And is this because their toxins are chemically different? Smaller, maybe?
Often, yes. Poisonous toxins are typically smaller chemical molecules, things like alkaloids or other potent organic compounds.
Hmm.
These smaller molecules can sometimes pass through skin membranes more easily, or they're just designed to be effective once they're inside the body, maybe absorbed through the gut or the lungs.
So the strategy for a poisonous animal is fundamentally different.
Very much so. While venom can be used for hunting or defense, poison is almost exclusively a defensive strategy. It's a way for the animal to protect itself by being, well, unpleasant or downright dangerous to eat or even touch.
Like a "don't eat me" sign.
Exactly. And many poisonous animals advertise this. They have bright warning colors. Scientists call it aposematism. It's like they're shouting, hey, I'm potentially toxic, so maybe look elsewhere for your lunch.
Right, the bright frogs and insects. Give us a few classic poisonous examples then.
Well, those bright poison dart frogs we mentioned are a prime example, secreting toxins through their skin. The pufferfish, of course, infamous for being deadly if it's not prepared exactly right because of the toxins in its organs.
Fugu.
Fugu, yes. And even some surprising birds or insects that become toxic if a predator tries to take a bite.
So that simple rule, bite it and you die, it's poison. If it bites you and you die, that's venom. It really works because it captures this fundamental difference in how the toxin gets in, driven by the toxin's chemistry and the animal's biology.
Right. That's precisely the key takeaway. It's all about the delivery. And, just to add a wild kind of theoretical note from the sources, stemming from this idea that venom needs a wound.
Yeah.
Technically, if you could somehow manage to drink snake venom without any cuts or ulcers in your mouth or throat, like zero openings.
Would you drink snake venom? Seriously?
Theoretically. And if your stomach acid was strong enough to break down those complex protein molecules before they could possibly be absorbed through the stomach lining, well, it might not be as harmful as getting it injected. Might, because the large protein molecules in venom would ideally just be digested like any other protein. But the sources really stress this is purely theoretical.
Okay, yeah, I'm sensing a massive disclaimer here.
Right.
We absolutely, under no circumstances, should anyone ever test this theory.
Oh, 1,000,000% no. Please, nobody try this. Any tiny scratch, any inflammation in your mouth, your esophagus, your stomach, that's enough to allow absorption. And even if it wasn't lethal, it could cause incredibly severe local tissue damage. It's just a scientific concept illustrating the delivery mechanism. Definitely not a survival tip.
Got it. Loud and clear. Safety first, always. Okay, point taken. Let's move beyond that basic distinction now and dive deeper into the sheer diversity of life that uses these strategies. Let's start with the passive defenders. Section 2, the diverse world of poisonous animals.
Right, and this is where things get really fascinating because a lot of poisonous animals don't actually make their toxins from scratch the way venomous animals often synthesize theirs. They acquire them.
Acquire them? How? From where?
Often, believe it or not, from their diet. The classic, most striking example is the poison dart frog.
Ah, yes.
These incredibly colorful frogs, they don't synthesize their potent alkaloid toxins themselves. They actually get them from eating specific insects and mites in their natural environment, particularly certain types of ants.
So they eat toxic ants.
They do, and somehow the frogs can eat these toxic prey items and then safely sequester or store the toxins in glands in their skin without being harmed themselves.
That's amazing. So if you raise a poison dart frog in captivity, say feeding it fruit flies or crickets,
it won't be poisonous.
Yeah.
Exactly. Because they lack that crucial dietary source of toxins, captive-bred poison dart frogs are generally harmless to touch. The sources specifically highlight studies showing this diet-dependent toxicity not just in poison dart frogs, but also in mantella frogs from Madagascar. They use the same clever strategy.
That's wild. An animal that's deadly dangerous in the wild could be harmless if raised differently. Are there other animals that get their poison from their food?
Oh, yes. It seems to be a fairly common evolutionary trick. Think about the monarch butterfly. Its caterpillars feed exclusively on milkweed plants. Milkweed contains these toxins called cardiac glycosides. The caterpillar is immune, but it stores the toxins. Then, the adult butterfly retains those toxins, making it taste absolutely foul, and it can actually make predators, like birds, quite sick if they try to eat one. It's a defense mechanism directly linked to their specific plant diet.
So the milkweed makes the butterfly poisonous.
In essence, yes. The sources also mention birds, like the spur-winged goose and the common quail, whose meat can apparently become toxic depending on what they've been eating, likely specific plants or maybe insects. And here's a real twist. Even some garter snakes can become poisonous.
Snakes. poisonous. I thought they were venomous.
Well, most garter snakes aren't dangerous, but if they eat rough-skinned newts, which are loaded with a really potent neurotoxin called tetrodotoxin, the snakes can absorb and store that toxin in their liver and other tissues. This makes the snake itself poisonous to potential predators, like birds of prey, that might try to eat it.
Wow, so you can become poisonous just by eating something poisonous. Nature is definitely metal.
It really is. It shows these complex ecological relationships, the movement of toxins through food webs. It's not always straightforward.
Okay, setting aside those diet-acquired toxins for a moment, let's look at some specific examples from the sources of animals that are poisonous through contact or ingestion, just by their own biology.
Right. So, beyond the diet-dependent frogs, many other frogs and toads simply secrete toxins from glands in their skin as a primary defense. Their bright colors are often that warning sign, that aposematism we talked about.
Stay away.
Exactly. Some, like the golden poison frog from Colombia, are incredibly toxic. The sources suggest that merely holding one could potentially be lethal because of the sheer amount of toxin absorbed through your skin. And many common toad species produce compounds called bufotoxins. The sources even note that some toads are intentionally milked for these substances, sometimes for traditional uses, sometimes for less savory purposes.
Any surprising poisonous mammals or birds? Mammals seem unlikely.
It's definitely rare in mammals, but the slow loris is often listed here. It has a gland on the inside of its elbow that produces a secretion. It licks this gland, mixes the secretion with its saliva, and then grooms itself, spreading this toxic mix onto its fur.
So if a predator bites it,
They get a mouthful of this irritating, toxic cocktail. Now, it also uses this toxic saliva in its bite, which is why some sources actually list it under venomous animals, too. It's kind of a hybrid case. Really interesting.
a bit of both.
Seems like it. As for birds, it feels counterintuitive, right?
Yeah.
But the hooded pitahui and the blue-capped ifrit birds, both from New Guinea, actually have toxins, specifically batrachotoxins, similar to the frogs, in their skin and feathers. Again, it's thought they acquire these from their insect diet, making them unappetizing or dangerous for predators.
Birds with toxic feathers. Okay, fish are another big category for poison.
Oh, definitely. The pufferfish is probably the most famous, containing that deadly tetrodotoxin, especially concentrated in organs like the liver, gonads, and skin. Preparing it as fugu requires incredible skill and licensing in Japan to remove those toxic parts safely.
I've heard about that. Very risky meal.
Extremely. Other fish mentioned include Greenland sharks and some barracudas. They can accumulate toxins like ciguatera through their diet, particularly as they get older and eat more affected reef fish. Eating the meat of these larger predators can cause serious illness.
And what about the invertebrate world,
hmm
especially in the ocean?
Oh, it's absolutely full of poisonous critters. Certain species of sea cucumbers can defend themselves by expelling sticky, toxic threads or secreting toxins into the water. Some starfish species are known to be toxic if eaten, and many other marine invertebrates, like some crabs, gorilla crabs are mentioned, and even some corals or anemone relatives, like certain Palaethoa species, contain potent toxins that make them dangerous if they're handled improperly or ingested.
Now, this is where my brain starts to bend a little. Are there any animals that manage to be both venomous and poisonous? They can inject you, but they're also toxic if you try to eat them.
Yes. This is a rare but super interesting double threat category. It's incredibly cool from a biological perspective. The sources highlight two prime examples. First, the blue-ringed octopus.
Ah, the tiny one with the bright blue rings.
That's the one. Beautiful, but incredibly dangerous. They have a potent, often fatal, venomous bite delivered by their beak-like jaws, used mainly for hunting crabs. But they're also poisonous if you were to eat them. Like those arrowworms we might mention later, they don't actually produce their main toxin, that infamous tetrodotoxin, again themselves. They get it from symbiotic bacteria living inside their salivary glands and other tissues.
So the toxin is just present throughout their body.
Exactly. It's in their tissues, making them dangerous even after they're dead, if ingested. There's a chilling anecdote in the sources about sea turtles accidentally eating them while foraging, becoming paralyzed by the ingested toxin, and drowning. Really underscores the potency of this dual threat.
Wow, a tiny octopus taking down a sea turtle just by being eaten. That's intense.
It's a powerful illustration of how these toxins function ecologically. The second dual threat animal mentioned is the Asian tiger snake, also known as Yamakagashi Rhabdophis tigrinus.
Another snake.
Yes, but with a twist. This snake has a venomous bite delivered by small fangs located towards the back of its mouth, what we call a pistoglyphus. Its venom can cause serious, sometimes fatal, bleeding problems, but it also has this unique secondary defense.
What's that?
It eats poisonous toads. And like the garter snakes with newts, it stores the toad's toxins, pufadienolide, in specialized glands in its neck called neutral glands. If a predator grabs the snake near the head, it can release these stored toxins from the neck glands as a deterrent.
So an animal that bites you with venom and can make you sick if you eat it, or even just grab it wrong because it's absorbed toxins from its own food, that's two completely different toxic strategies in one creature.
It's a remarkable adaptation, isn't it? It showcases the complex evolutionary pressures that drive the development of such diverse defensive and predatory tools. It really does blur the lines between venom and poison in a fascinating way.
Okay, let's transition now to the other side of the coin. Let's delve deeper into the world of venomous animals, focusing specifically on how they actually deliver those toxins. Section 3, the diverse world of venomous animals.
Right. As we've established, venom delivery isn't passive. It requires a specific piece of biological kit, you might say, a specialized apparatus to get the job done.
And these delivery tools vary a lot across different animal groups.
Absolutely. While fangs and snakes and spiders might be the most iconic, the sources list a whole range of tools. You've got stingers and scorpions, bees and wasps, sharp modified spines in fish or sea urchins, and in some rare surprising instances, even modified teeth or claws, like in the platypus or the slow loris we just talked about.
Okay, let's start with the classics then. Arachnids, spiders and scorpions.
Right. Technically, the sources point out that pretty much all spiders have venom. They deliver it through their fang-like mouthparts called chelicerae. However, the vast majority are completely harmless to humans. Their fangs might be too small to pierce our skin, or their venom just isn't potent enough to cause us any significant harm.
But there are definitely some we need to worry about.
Oh, for sure. The sources highlight the groups that are medically significant or dangerous. You've got the funnel web spiders, especially those from Australia, the Brazilian wandering spiders, known for being aggressive, all the widow spiders, black, brown, red, the Australian redback. They're often recognized by that distinct red or orange hourglass marking on the underside of their abdomen.
All right, the hourglass.
And then the recluse spiders, like the brown recluse with its characteristic violin-shaped marking on its back, and the related Chilean recluse. Tarantulas also have venom delivered via fangs, though their bites are usually less severe than the others mentioned. But some also have another defense, flicking irritating hairs, called urticating hairs, from their abdomen.
Hairs that sting. Ugh. Okay, and scorpions. Do all scorpions sting?
Yes, all known scorpion species possess venom, which they deliver via that stinger, the telsin, at the very end of their segmented tail. Again, though, while all are venomous, only a relatively small fraction, maybe a few dozen out of over 2,000 species, have venom potent enough to be seriously dangerous or lethal to humans.
Which ones are the most dangerous?
The sources list some infamous examples. Things like the bark scorpions, found in the U.S., Southwest, and Mexico. The deathstalker, Lyrus quinquestriatus, from North Africa and the Middle East. Various species from genera like Titius in South America, Androctonus, the fat-tailed scorpions, in North Africa and the Middle East, Parabuthus in Southern Africa, and Hottentotta across Africa and Asia. Many of the most dangerous ones are found in arid or desert regions.
Got it. Moving into the insect world and other arthropods. Well, bees and wasps are definitely well known for their stingers.
Precisely. Bees, wasps, and also certain ants use stingers for venom delivery. Interestingly, in bees and wasps, the stinger is actually a modified ovipositor, the egg-laying structure, which is why only females can sting. Male bees and wasps don't have one.
Ah, that explains it.
Some caterpillars also possess those urticating hairs we mentioned, with the tarantulas tiny, brittle hairs that break off easily and can inject irritating or sometimes quite toxic compounds upon contact. Think of certain tussock moth caterpillars. And the bullet ant, Paraponera clavata, from Central and South America, is singled out for having arguably the most painful insect sting known to science, though it's usually not fatal unless someone has a severe allergic reaction, like anaphylactic shock.
Just incredibly painful.
Rated highest on the Schmidt Sting Pain Index, apparently. Beyond insects, even centipedes can inflict a venomous bite using modified legs called forcipules near their head. And the sources mention this really fascinating discovery of a unique venomous crustacean, Cebulbanus tulumensis, a type of remipede found living in underwater caves, the cenotes, in Mexico's Yucatan Peninsula.
A venomous crustacean, like a shrimp or crab relative. You weren't kidding about diversity.
It seems venom has evolved in almost every corner of the animal kingdom.
What about marine animals? The ocean seems like a chemical weapons zone sometimes.
It really can be. Take cone snails, for instance. These mollusks have beautiful shells, but they are highly efficient predators. They hunt using a modified tooth, a radular tooth, that's shaped like a tiny harpoon.
Harpoon.
Yeah, they fire this harpoon connected by a thread into fish or other prey, and through it they inject this incredibly complex mix of neurotoxic peptides called conopeptides. These paralyze the prey almost instantly. Some species are deadly to humans. Octopuses, squid, and cuttlefish also use venom, typically delivered via a bite from their beak, primarily for hunting. And remember, our dual-threat blue-ringed octopus fits here, too.
Right, and then there are things that just sting you if you accidentally brush up against them in the water.
That would mainly be the cnidarians. This group includes jellyfish, sea anemones, corals, and the Portuguese man-o'-war, which looks like a jellyfish but is actually a colony of organisms. They all have these specialized stinging cells called nematocysts, usually concentrated on their tentacles or tissues. Think of them like tiny, coiled springs inside a capsule, each with a barbed thread and a payload of venom at the tip. When triggered by touch or chemical cues, the capsule bursts open and the barbed thread fires out like a microscopic harpoon, injecting venom into the unfortunate victim. It's an incredibly fast and efficient mechanism.
Ouch. Anything else stinging in the sea?
Some echinoderms, like certain types of sea urchins, also have venomous spines for defense, or they might have tiny pincer-like structures scattered among the spines called pedicelaria, which can also grasp and inject toxin.
Those arrowworms we talked about earlier, the ones getting toxins from bacteria, are they considered venomous too?
Yes, the chitonathans, or arrowworms. They use grasping spines near their mouth to seize prey and inject toxins. And like the blue-ringed octopus, they utilize tetrodotoxin that seems to come from symbiotic bacteria. So it's a different delivery apparatus grasping spines, but still involves injection using an acquired toxin. And even some segmented worms, the annelids, like bloodworms, Glycera species, possess venom glands connected to their jaws used to subdue prey.
This list is getting huge. It's way more widespread than just snakes and spiders. What about vertebrates, then, beyond snakes? Are there other fish, amphibians, reptiles, or even mammals that are truly venomous?
Absolutely, and some are quite surprising. Fish are actually a massive category for venom. Estimates in the sources suggest at least 1,200 venomous species, possibly many more. Many have venom glands associated with sharp spines on their fins, like dorsal, pectoral, or anal fins, or sometimes near their gills.
What are some well-known venomous fish?
The stonefish, Synanthia species, is notoriously painful and often called the most venomous fish in the world. It delivers potent neurotoxins and other compounds through stout spines on its back, which can easily puncture a foot if stepped on. Lionfish, pterois species, with their flamboyant fins, are also venomous, as are many related scorpion fishes. And many species of catfish possess venomous spines, usually one behind the dorsal fin and one behind each pectoral fin, used purely for defense. Getting stung by a catfish spine can be surprisingly painful.
I'll watch out for catfish spines, then. Any venomous lizards? I know about Gila monsters.
Yes, the Gila monster and its close relative, the Mexican beaded lizard, are the most well-known venomous lizards. They have venom glands in their lower jaws, and the venom flows along grooves in their teeth as they bite and chew. Recent research, using methods like proteomics to analyze saliva, suggests that some larger monitor lizards, including the famous Komodo dragon, as well as the Parentian lace monitor in Australia, also possess complex venom components in their saliva. Though their delivery system isn't as specialized or efficient as a snake's fang system, it might play a role in subduing prey or digestion.
Interesting. Komodo dragons, too. And amphibians. They're usually poisonous secreting toxins, aren't they?
Mostly poisonous, yes, that's their main toxic strategy. But a few are considered truly venomous, meaning they have a specific mechanism to inject the toxin, not just secrete it. The sources mention Bruno's casque-headed frog and Greening's frog from South America. These frogs have bony spines on their heads that are associated with poison glands. They can apparently deliver toxins defensively by headbutting predators.
Headbutting venomous frogs.
It sounds bizarre, doesn't it? Fire salamanders and Iberian ribbed newts also have mechanisms the newt can actually push its ribs through its skin, and the ribs get coated with toxin from skin glands, acting like venomous spines. And in a really surprising recent discovery, even caecilians, those legless, burrowing amphibians that look kind of like large worms or snakes, have been found to possess specialized glands near their teeth, possibly producing venomous secretions delivered through grooved teeth, similar in structure to some snake venom glands.
That is completely unexpected. Caecilians might be venomous, and mammals, You said it's rare, but they exist.
It is rare in modern mammals, yes, but some fascinating examples exist. The male platypus is famous for having sharp, venomous spurs on its hind ankles. The venom is potent enough to cause excruciating, long-lasting pain in humans and can kill smaller animals like dogs. It seems to be primarily used during the breeding season for competition between males.
Ouch. Platypus spurs.
What else? Several species of shrews, including the northern short-tailed shrew found in North America, have venomous saliva delivered via grooved incisors, used to paralyze insect and small vertebrate prey. Selenodons, which look somewhat like large shrews and are found only in Cuba and Hispaniola, are also venomous mammals with toxic saliva. And the slow loris, which we discussed under poisonous animals for its toxin-laced fur, also delivers a toxic bite using its unique saliva mixed with the arm gland secretion. So it firmly belongs in the venomous category too, possibly even primarily. The pygmy loris is also noted as venomous. The sources suggest this trait might have been more common in ancestral mammals and has been lost in most lineages.
It's truly staggering how many different ways life has figured out how to inject toxins across so many different branches of the tree of life, from snails to shrews.
It really speaks volumes about the evolutionary advantages of having such a potent tool, whether it's for catching food more efficiently or for defending yourself effectively against becoming food.
And perhaps the most iconic, most sophisticated example of a venom delivery system belongs to snakes. So let's do that deep dive we promised, specifically into their biting and injecting apparatus. Section 4. A deep dive into snake venom systems.
Right. Snakes have really perfected this biological weapon system. It's this incredibly integrated system involving glands, ducts, specialized teeth called fangs, plus muscles and bones, all working together in precise coordination.
So what are the main components you'd find in a venomous snake's head?
The sources describe it as essentially consisting of three main parts: a pair of modified salivary glands that actually produce the venom mixture, ducts, which are tubes that carry the venom forward from the glands, and the fangs themselves, which are those specialized teeth designed for injection.
Okay, glands, ducts, fangs.
Right.
Where exactly are these glands located?
The venom glands are typically sac-like structures situated on either side of the upper jaw, towards the back of the head, below and behind the eye. They're often thought to be modified versions of the parotid salivary glands found in non-venomous reptiles and mammals. From each gland, a narrow tube, the venom duct, runs forward along the upper jawbone and connects to the base of the fang. These glands are held securely in place by a network of connective tissue and ligaments.
And the fangs themselves, what makes them different from regular teeth?
Fangs are basically enlarged teeth, usually on the maxilla, upper jawbone, that are very sharply pointed and often hook-like. They're adapted for piercing skin and injecting venom. They are incredibly tough, calcified structures covered in enamel like other teeth. An important detail is that snakes can, and regularly do, regenerate fangs if they break off or are shed, ensuring they almost always have a functional injection tool ready.
That's handy. Now, the sources get into different types of fangs based on their structure and how they're positioned in the mouth. Can you simplify that bit for us?
Yeah, it can seem complicated, but you can break it down. Structurally, think about how the venom travels along or through the fang. Some are like an open gutter or groove running down the front surface. This is often called the open type, or just grooved. You see this in snakes like cobras. Others have evolved so that the groove has folded over and fused, forming a hollow tube, much like a hypodermic needle. This is the closed type, or tubular fang, typical of vipers. This closed tube allows for more efficient, higher pressure injection.
Okay, open groove versus closed tube. And then there's the position, how they actually sit in the mouth.
Right, and this often correlates with different snake families and their evolutionary history. There are generally four main functional types described. First, a glyphus. This literally means without grooves. These are snakes that have no specialized fangs at all. They just have rows of solid, usually similar-sized teeth used for gripping prey while swallowing. Think of pythons, boas, most common non-venomous snakes like garter snakes or rat snakes.
Yeah.
They are not venomous in the injecting sense.
Got it. No fangs, no venom injection system. What's next?
Second, opistoglyphus. Opisto means behind. Glyph means groove or hollow. These snakes have grooved fangs, but they are located near the back of the upper jaw, behind the regular teeth. These are often called rear-fanged snakes. Examples include the boomslang, vine snakes, the Asian tiger snake we mentioned earlier. Their venom glands, sometimes called Duvernoy's glands, are often less developed and aren't typically enclosed in strong muscles for high-pressure injection like front-fang snakes. Venom tends to seep into the wound as the snake chews or holds on, rather than being actively squirted in a quick strike. The venom pressure is relatively low, maybe less than 5 psi according to some sources.
So a less direct, maybe slower delivery method. Needs to chew a bit. What about the ones with fangs right at the front?
Okay, now we get to the front-fanged snakes. There are two main types here. Third is Proteroglyphus. Protero means first or front. Their fangs are located at the front of the upper jawbone, maxilla. These fangs are generally shorter and thicker than the next type we'll discuss, and they are often relatively fixed in place, pointing mostly downwards, though some have limited mobility. This group includes elapids like cobras, kraits, mambas, coral snakes, and sea snakes. They may have other small teeth behind the fang, but there's usually a distinct gap, a diastema. Venom delivery here is much more efficient than in rear fang snakes. Pressure on the venom glands can exceed 30 psi, allowing for rapid injection during a quick bite.
And you mentioned spitting cobras have a modification here.
Yeah, a really cool adaptation. Spitting cobras have a modification near the fang tip. The exit hole, or orifice, is smaller and rounder and angled forward. This allows them to forcefully spray or spit venom quite accurately towards a perceived threat's eyes as a defensive measure rather than needing to bite.
That spitting is amazing. Okay, fixed front fangs for proteroglyphs. And finally, the ones with the really long folding fangs.
That's the fourth type, Selenoglyphus. Seleno means pipe or channel. These snakes, found in the Viperidae family, vipers, rattlesnakes, pit vipers, have the most highly developed venom delivery system. They possess incredibly long, hollow, tubular fangs like hypodermic needles. These fangs are the only teeth on a greatly reduced maxilla bone, which is hinged and can rotate. This hinge mechanism allows the fangs to fold back against the roof of the mouth parallel to the jawline when the mouth is closed, but when the snake strikes...
They swing forward.
Exactly. The maxilla rotates, swinging the fangs forward and downward, sometimes nearly 90 degrees, into a perpendicular striking position, like twin switchblades. This allows for deep penetration into the prey or target. The hollow core of the fang connects directly to the venom duct, and this system allows for very efficient high-pressure venom injection, again, often exceeding 30 psi. It's arguably the pinnacle of snake venom delivery evolution.
That image of the fangs swinging forward like switchblades is really vivid and kind of terrifying. What about the muscles and bones that make all this happen? It sounds complicated.
It is a complex interplay of muscles and bones working in perfect sync. You have muscles like the digastric that attach the skull to the lower jaw, helping to pull the mouth open very wide. Then other muscles, like the sphenopterygoid, pull on a chain of bones in the palate, the pterygopalatine transverse arch, causing the maxilla, the fang-bearing bone, to move or rotate forward, thus erecting the fangs. This is particularly dramatic in the selenoglyph vipers with their true rotary maxilla.
And then muscles to close the jaw and squeeze the glands.
Right. Powerful temporal muscles, located on the sides of the head, contract to forcefully close the jaws, driving the erected fangs into the target. As the fangs embed, other muscles, often parts of the same temporal muscles in some groups, or specialized compressor muscles associated directly with the gland in vipers, squeeze the venom glands like squeezing a bulb, forcing the venom out through the ducts and fangs. The sources describe it as a rapid, highly coordinated sequence of events.
The sources break down the entire biting sequence into 4 quick phases. Can you walk us through those briefly?
Sure. Phase 1 is the strike itself, a rapid forward movement of the head and neck towards the target. Snakes can strike incredibly fast, often covering up to a third of their body length. Vipers are particularly known for their speed here. Some snakes, like cobras with less mobile fangs, might raise the front part of their body to get a better angle.
Okay, phase 2.
Phase 2 is opening the mouth and fang elevation. As the snake nears the target, the lower jaw is depressed, open wide, by muscles like the digastric. Simultaneously, muscles pulling on that palatal bone chain cause the maxilla to move, either sliding forward and upward slightly in proteroglyphs or rotating dramatically forward in selenoglyphs, swinging the fangs into their functional striking position. The degree of elevation varies from maybe 10-15 degrees in some to 45-50 degrees or even more in vipers.
Fangs deployed. Phase 3.
Phase 3 is closure and venom injection. Once the fangs make contact and start to penetrate the target, the powerful temporal muscles contract, closing the jaws forcefully. This drives the fangs deeper. At the same time, or fractions of a second later, the muscles surrounding the venom gland contract, compressing the gland and expelling the venom through the ducts and out the fang tips into the tissue. This happens incredibly quickly.
Injection complete. And the final phase.
Phase 4 is retraction. After injecting the venom, retractor muscles pull the fangs downward and backward, disengaging them from the wound. The snake then withdraws its head, ready to either wait for the venom to take effect on prey or to escape from the danger it was defending against.
It's just remarkable how rapidly all of those intricate steps happen in sequence. A perfectly timed biological mechanism.
It really is. It's a highly evolved, incredibly efficient system for delivering a potent chemical payload quickly and effectively, often taking only fractions of a second from the start of the strike to the completion of injection, especially in those advanced vipers.
Before we move on from snakes, the sources also provide some physical differences that sometimes distinguish venomous from non-venomous snakes. Now, we should absolutely stress that you should never rely solely on appearance to identify a snake and always exercise extreme caution. But are there some general visual cues mentioned?
Yes, there are some general patterns often cited, but it's absolutely crucial to remember there are many exceptions, and identification should always be left to experts. That said, generally speaking, in many regions, venomous snakes like vipers might have a head shape that appears more distinctly triangular or arrowhead-shaped when viewed from above, broader than the neck, compared to the typically more slender, spoon-shaped, or rounded head of many common non-venomous snakes. Also, the scales on the top of the head in vipers tend to be small and numerous, almost granular, whereas many non-venomous snakes, and also elapids like cobras, have large, plate-like, symmetrical scales covering the head.
Okay, head shape and head scales. What about their eyes? Heard about pupil shape.
That's another commonly mentioned, though again not absolute, difference. Many venomous snakes, particularly pit vipers, have pupils that are vertically elliptical or slit-shaped, like a cat's eye in bright light. Many non-venomous snakes, and also venomous elapids like cobras and coral snakes, typically have round pupils. However, this rule has many exceptions worldwide. Some dangerously venomous snakes have round pupils, and some harmless snakes have elliptical ones, so it's definitely not a foolproof identifier on its own.
Not foolproof, got it. Any other body shape or scale pattern sometimes mentioned?
Some venomous snakes, particularly vipers, tend to have a stouter, thicker body relative to their length compared to the more slender build of many non-venomous snakes, but again, this is very generalized. A potentially more reliable but still not universal sign involves looking at the scales on the underside of the tail after the vent or anal plate. In many non-venomous snakes, the scales here, subcaudal scales, are arranged in a double zigzag row. In many venomous snakes, particularly vipers and elapids, these scales form a single row, at least for part of the tail length. The broad belly scales, ventral pleats that span the entire width of the body, are typically present in both groups.
Single versus double row under the tail. Interesting. And lastly, if you are unfortunate enough to be bitten and could safely look at the bite mark.
This is perhaps the most telling sign after the fact, assuming venom was actually delivered. A bite from a non-venomous snake will typically leave just rows of small, fairly uniform teeth marks, maybe in a horseshoe shape. A bite from a front-fanged venomous snake, proteroglyph or solenoglyph, will usually leave one or two distinct, often deeper puncture marks from the fangs, possibly in addition to other smaller teeth marks. Rear fang snakes might leave fang marks further back, or the marks might be less distinct.
Fascinating distinctions in anatomy and appearance, but absolutely point taken on not getting close enough to try and do a scale count or check pupil shape in the field.
Definitely not. The safest approach is always to give any snake plenty of space, admire it from a very safe distance, and assume it could be venomous unless you are absolutely certain otherwise, based on expert identification for your specific region.
Right. Wise advice. Okay, so we've explored the danger, the different strategies, and the incredible biological mechanisms involved. Now let's pivot to the really surprising turn in this story. Section 5. Beyond the danger, the healing power of toxins. It just seems so counterintuitive, doesn't it? That these substances evolved specifically to cause harm are proving incredibly valuable in medicine.
It really does seem like a paradox, but it's where the story gets truly fascinating. These incredibly complex cocktails of molecules, which nature has refined over millions of years to target very specific biological processes in prey or predators. Well, they're turning out to be incredibly precise tools for medical research and drug development.
Was this idea of using venoms in medicine something people thought of historically? Or is it purely modern?
No, the sources mention surprisingly ancient uses. It wasn't just modern science that saw potential here. In places like ancient Rome and Greece, various animal venoms were apparently incorporated into remedies or complex mixtures intended to treat all sorts of ailments, from things like smallpox and fever to wound healing. And the famous theriac, this incredibly complex medicinal mixture that was used for centuries across Europe as a kind of cure-all or universal antidote, often contains snake venom as one of its many ingredients.
Theriac. I've heard of that. Containing snake venom. And the modern medical era really started with antivenoms, didn't it? Treating the bites themselves.
Exactly. That was the first major modern breakthrough. It came in the late 19th century with the work of Albert Calmette, a French scientist. He pioneered the development of antivenoms by injecting small, carefully controlled, non-lethal doses of venom into animals, usually horses. Their immune systems would then produce antibodies specifically targeted against the venom components. Harvesting these antibodies from the horse's blood serum gave us the first effective way to neutralize the effects of a snake bite in a human victim. It was revolutionary.
A huge step. But then scientists started looking beyond just neutralizing the venom.
Yeah.
They started looking at the components of the venom itself for other uses.
Right. That was the next logical step. A really key moment highlighted in the sources was a discovery back in 1934. Researchers found that cobra venom, when used in very tiny controlled amounts, had incredibly powerful pain-relieving analgesic properties, reportedly much stronger than morphine, the standard at the time, and significantly, it didn't seem to cause the same kind of addiction or tolerance issues associated with opiates. This really opened scientists' eyes to the potential of individual venom toxins as sources for novel therapeutic drugs.
Stronger than morphine, without addiction. That sounds like a holy grail for pain relief. I know I've seen some traditional remedies or maybe ointments containing snake venom still being sold today in some places.
They are, yes. You can still find creams or balms claiming to contain snake or bee venom, often marketed for arthritis or muscle pain. However, the sources generally point out that the scientific evidence for their effectiveness is often limited or questionable. The actual amount of venom might be minuscule, the active components might not penetrate the skin effectively, and any perceived benefit could be due to other ingredients, a placebo effect, or just the rubbing action itself. The real power of venoms in modern medicine comes from isolating specific, active molecules and understanding precisely how they work at a molecular level.
So, isolating the active ingredients. Where are venoms making a real impact today in modern science and medicine?
In several key areas, really. First, as fundamental tools in basic research. Because venom components have evolved over millennia to bind very specifically and tightly to certain targets in the body, things like ion channels, receptors on cell surfaces, or critical enzymes, they are incredibly valuable probes for scientists trying to study how those biological processes work.
Can you give an example?
Sure. A classic one is alpha-bungarotoxin, a potent neurotoxin from the venom of a crate snake. It binds extremely selectively to a specific type of acetylcholine receptor, the nicotinic type found at nerve muscle junctions. For decades, it's been an unsurpassed tool for researchers studying how nerves communicate with muscles. Similarly, alpha-conotoxins from cone snails are invaluable for dissecting the different subtypes of nicotinic receptors in the brain. Toxins from scorpions and spiders are highly prized for their selectivity in studying various types of ion channels, which control electrical signaling in nerves and other cells. And numerous venom proteins that affect blood clotting have been crucial for understanding the complex cascade of steps involved in hemostasis.
So they're like highly specific molecular keys used by researchers to unlock the secrets of how our cells and bodies function at the most basic level.
That's a perfect analogy. They target a specific molecular lock, often better than any synthetic chemical can, allowing researchers to understand that specific lock's function in detail.
Okay, research tools. What about diagnostic applications? Using venom to help diagnose diseases.
Yes, this is another important area. Several standard diagnostic tests used every day in clinical laboratories around the world are actually derived from snake venoms. For instance, the sources mention tests developed by Penapharm using specific venom components. Acherin, from the saw-scaled viper, Achis carinatus, is used in the Acherin clotting time test to detect abnormal prothrombin or monitor certain anticoagulants. Protashi, derived from the southern copperhead, Achistrodon contortrix, is used in assays to determine levels of protein C and protein S, important natural anticoagulants in our blood. And RVVX, an activator of factor X from Russell's viper, Daboia russelli venom, is used in tests for factor X deficiency and also as a key region in screening tests for lupus anticoagulants, which are antibodies that can cause clotting problems.
That's incredible. Components from a deadly snakebite are now part of routine blood tests, helping diagnose clotting disorders. What about biotechnology uses?
In biotechnology, compounds derived from venom are being explored or used for various applications. The sources mention, for example, a fibrin adhesive or sealant developed from the venom of a South American rattlesnake, Crotalus starificus. This has been studied as a potential biological glue to seal tissues during surgery, perhaps as an alternative to sutures in some situations. Other specific enzymes isolated from venoms, like certain highly specific proteases, protein-cutting enzymes, are sometimes used as tools in molecular biology for processes like cleaving recombinant proteins during purification.
And then there's the really big one, drug development. Actually taking these toxins or blueprints based on them and turning them into medicines people can take.
This is arguably where venoms have had their most profound impact on human health. And it's where the concept of rational drug design, using knowledge of biological targets to create drugs, was really pioneered, thanks in large part to venom research. By identifying a venom component that does something very specific in the body, say, dramatically lowers blood pressure, or prevents blood clotting, or blocks a specific pain signal, scientists can use the structure of that natural toxin as a starting point, a template, to design a new drug that mimics that beneficial effect but is safer, more stable, and controllable for therapeutic use.
What are the most famous examples, the real success stories of drugs that originated from venom?
The absolute blockbuster, the superstar example, is definite captopril, and its later generation successors like enalapril and lisinopril. These are among the most widely prescribed drugs in the world for treating high blood pressure, hypertension, and heart failure. Their development was directly based on identifying peptides in the venom of a Brazilian pit viper, Bothrops jaraca.
How did that work? What did the viper peptide do?
This specific peptide was found to potently inhibit an enzyme in the body called ACE angiotensin-converting enzyme. ACE plays a key role in producing angiotensin II, a substance that normally constricts blood vessels and raises blood pressure. By blocking ACE, the viper peptide caused a rapid drop in blood pressure in prey animals. Scientists realized they could design a drug that mimicked this ACE-inhibiting action. The development of captopril in the 1970s was a landmark achievement, validating this approach and leading to a multi-billion-dollar class of drugs that has saved countless lives.
So a snake venom peptide that causes a dangerous drop in blood pressure in prey became the blueprint for a drug that safely treats high blood pressure in millions of humans. That's absolutely fascinating.
It is a perfect example of turning nature's weapon into a human medicine. Another major success story is in the field of blood thinners, or more accurately, antiplatelet drugs. Two important drugs used intravenously in hospitals to prevent heart attacks and dangerous blood clots, especially during procedures like angioplasty, are integrilin, generic name eptifibatide, and agrestati, turofibon. Both were developed based on proteins found in viper venoms.
Which vipers this time?
Eptifibatide is a synthetic peptide based on a protein called barberin, found in the venom of the southeastern pygmy rattlesnake, Cistris miliaris barburi. Turofibon is a small molecule drug designed based on the structure of disintegrins, a class of proteins found in the venom of snakes like the saw-scaled viper, Achis carinatus. These venom components interfere with a key receptor on platelets, the GPI-beta-EI receptor, that allows them to stick together and form clots. So toxins that likely evolved to cause bleeding or clotting problems in prey animals were used as the basis for drugs that prevent unwanted, dangerous clotting in cardiovascular patients.
That's another brilliant reversal, using something that disrupts normal clotting to prevent dangerous clots and disease states. What about pain relief? You mentioned that early cobra venom finding. Has anything come from that?
While a drug based directly on that specific cobra toxin hasn't become a mainstream painkiller yet, the search for venom-derived analgesics is very active. And we do have a success story from the marine world. The drug Prelty, generic name is iconotide, is a synthetic version of a peptide toxin, omegaconotoxin MVIA, originally isolated from the venom of the magician cone snail, Conus magus.
A cone snail peptide for pain. How does it work?
This peptide is a highly selective blocker of a specific type of calcium channel, N-type, found in nerve cells that transmit pain signals. By blocking these channels in the spinal cord, Prelt can provide significant relief from severe chronic pain. It has to be delivered directly into the spinal fluid via an implanted pump, so it's reserved for patients with very severe pain who haven't responded to other treatments, like opioids, or who can't tolerate their side effects. But it represents another unique mechanism of action derived directly from a venom toxin.
Amazing. From a snail's harpoon to a potent pain drug. The sources also share that really striking story about the poison dart frog toxin, the template for ABT 594. Can you account that?
Yes, it's a really important anecdote that highlights a different aspect of this field. Researchers at Abbott Laboratories were developing a promising new painkiller candidate called ABT 594. It was based on the chemical structure of epibetadine, a potent analgesic compound isolated from the skin of a specific species of Ecuadorian poison dart frog, Epipedobates tricolor. This compound worked differently from opioids, targeting nicotinic acetylcholine receptors involved in pain signaling, and it showed potential as a powerful, non-addictive pain reliever.
Sounds great. What was the problem?
The problem was that the specific small area in Ecuador where the original frogs were collected for study was tragically destroyed, cleared for banana plantations. The researchers nearly lost access to the natural source of this incredibly valuable chemical blueprint before they had fully characterized it or secured alternative supplies.
Wow, a potential breakthrough painkiller almost lost because of habitat destruction.
Exactly. Fortunately, the story has a somewhat happier ending. They were eventually able to find the same frog species in another location and continue their research, although ABT 594 itself ultimately faced challenges in clinical development due to side effects. But the near miss serves as a powerful, real-world example of how biodiversity loss isn't just about losing species in an abstract sense. We are potentially losing access to unique, undiscovered biological compounds, chemical innovations perfected by evolution that could hold the key to future medicines, research tools, or other scientific breakthroughs we can't even imagine yet. It really underscores the immense untapped value of conserving these complex ecosystems and the incredible chemical diversity within them.
That story really brings home the critical link between conservation efforts and potential medical progress. It leads perfectly into our final section, actually, Section 6. The frontier prospects and challenges in venom research. Where is this exciting field headed next, and what are the major hurdles or challenges still facing researchers?
Yeah, the field is definitely not standing still. It's evolving rapidly, largely thanks to the development of powerful new technologies that let us probe the complexity of venoms in ways that were just impossible even a decade or two ago.
You mentioned the omics technologies earlier. Proteomics, transcriptomics.
Precisely. These high-throughput techniques are revolutionizing the study of venoms. Proteomics allows scientists to identify and often quantify virtually all the proteins and peptides present in a single venom sample. Transcriptomics lets them identify all the messenger RNA molecules being produced in the venom gland. These are essentially the blueprints, the instructions transcribed from DNA, that tell the gland which toxin proteins to make. And genomics, sequencing the animal's entire DNA, can reveal the genes themselves that code for these toxins. When these omics approaches are applied specifically to studying venoms, the field is often called venomics.
So Venomics gives a complete picture of the venom.
It aims to, yes. It allows us to get an incredibly detailed inventory of the chemical arsenal within a venom. We can identify hundreds, sometimes even thousands, of different molecules in a single venom sample, including compounds that are present only in very tiny amounts, low abundance, that would have been missed by older methods. This helps us understand exactly what each venom is made of, which components are likely responsible for its toxic effects, how those components might work together synergistically, and even how venom composition can vary, say, between individuals, between different geographic populations of the same species, or even change with the animal's age or diet.
That detailed knowledge must be incredibly useful.
It is. The sources mention, for example, using venomics to discover entirely new types of enzymes, metalloproteinases, in the venom of a rear-fanged snake, Thamnodonastes strigatus, that hadn't been seen before. Sequencing the entire genome of the king cobra provided a complete catalog of its venom toxin genes, shedding light on their evolution. This detailed molecular knowledge is the foundation for much of the future work in the field.
What are some particularly exciting areas researchers are focusing on right now using these tools?
One very promising but also very challenging area is the continued search for venom components that can act on the central nervous system, CNS, the brain, and spinal cord. The goal is to find new treatments for complex conditions like chronic pain, epilepsy, Parkinson's disease, Alzheimer's, maybe even psychiatric disorders. Many venom toxins are known to target ion channels and receptors that are crucial for nerve cell function, so the potential is definitely there.
The big challenge there is getting drugs past the blood-brain barrier, right? That protective shield around our brain.
Exactly. The blood-brain barrier, BBB, is a major hurdle for getting many potential drugs, especially larger molecules like peptides or proteins, into the brain where they need to act. It's designed to keep harmful substances out. However, excitingly, research is showing that some venom peptides can naturally cross the BBB, or can be engineered to do so. The sources mention that certain peptides from bee venom, like an analog of apamin, and scorpion venom, notably chlorotoxin, have demonstrated this ability to get into the brain.
And there's a real-world application already emerging from this ability.
Yes, a really cutting-edge one. The sources highlight that scorpion chlorotoxin has this remarkable dual property. It can cross the blood-brain barrier, and it seems to selectively bind to certain types of cancer cells, particularly glioma brain tumor cells, more than normal brain cells. Researchers have cleverly exploited this by linking chlorotoxin to a fluorescent dye. This conjugate molecule is called tumor paint, BLZ-100.
Tumor paint.
Yes. When injected intravenously before or during surgery, it crosses the BBB, binds to the tumor cells, and makes them glow under near-infrared light. This allows surgeons to visualize the boundaries of the cancer much more clearly, helping them to remove more of the cancer's tissue while sparing healthy brain tissue. It's currently in clinical trials and shows incredible potential for improving brain cancer surgery outcomes. It's a fantastic example of turning a scorpion toxin into a precision tool for fighting cancer.
That is truly amazing, a scorpion toxin literally lighting up cancer cells to guide the surgeon's hand. Science fiction becoming reality indeed.
It really is, and it's powered by understanding the unique properties of a venom component. Beyond that specific example, research is ongoing by various biotech companies, mentioned in the sources, to develop new drugs based on known toxins or modified versions for various diseases, including cancer, using cratoxin cardiotoxin from rattlesnake cobra, neurological diseases, modified cobra toxins, and autoimmune diseases like arthritis and psoriasis, using an anemone toxin derivative. But the sources also caution that this is a high-risk, competitive, and expensive area of research. Even well-characterized toxins can sometimes have unexpected effects when tested in whole organisms in vivo.
So, lots of promise, but still challenging. How are these new technologies helping with the original fundamental medical need better antivenoms for treating bites and stings?
Hugely important application. Antivenom therapy, using antibodies raised in animals like horses or sheep, remains the primary, life-saving treatment for serious envenomation by snakes, scorpions, spiders, etc. Understanding the precise protein composition of a particular venom using omics technologies is absolutely critical for developing the most effective antivenoms.
Why is knowing the exact composition so important?
Because you need to ensure that the antivenom contains neutralizing antibodies against all the medically important toxins present in the venom of the species found in a specific region. Venom composition can vary significantly even within the same snake species, depending on its geographic location, age, or even recent diet. If the antivenom was made using venom from snakes from a different region, it might not effectively neutralize all the key toxins in a local bite, leading to treatment failure. Venomics allows for precise characterization of regional venom variations, guiding the production of more tailored and effective regional antivenoms.
Makes sense. Tailoring the antidote to the specific poison. Are there new ways emerging to actually produce antivenoms too?
Yes. One promising approach mentioned is DNA immunization. Now that transcriptomics can identify the exact genetic blueprints, cDNA, for the key toxins, researchers can use these blueprints directly. Instead of injecting the actual, potentially dangerous toxin protein into an animal, like a horse, to raise antibodies, they can inject the DNA or RNA that codes for the toxin. The animal's own cells then temporarily produce the toxin protein themselves, safely inside the body, triggering a strong immune response and the production of neutralizing antibodies. This method has shown promise, for example, in generating antibodies against key toxins from the venom of the highly dangerous pit viper Bothrops asper in Central America. It might eventually offer a safer, more controlled way to produce antivenoms.
So despite all this incredible progress in understanding venoms and finding uses for them, what are the big remaining challenges in this field? Where do we still need to make breakthroughs?
There are definitely still significant hurdles. One of the biggest remains the massive global need for more effective, more affordable, and more easily accessible antivenoms, particularly in the rural, impoverished areas of tropical countries in Africa, Asia, and Latin America, where snakebite is a major and often tragically neglected public health crisis, killing tens of thousands and disabling hundreds of thousands every year. Producing enough high-quality, appropriate antivenom and getting it to the people who need it most remains a huge challenge.
So access and affordability of treatment is still a major issue. Are there still venoms out there that we just don't understand well enough yet?
Absolutely. Far from it, in fact. The sources point out that for some extremely dangerous animals, the venom composition is still not fully characterized, and there's no well-established specific treatment beyond supportive care. A prime example given is certain species of Australian box jellyfish that cause the severe and sometimes fatal Irukandji syndrome. We still don't know exactly which toxins cause all the symptoms, and there's no specific antivenom. The same is true for the venoms of many medically significant scorpions and spiders around the world. So while we have these incredibly sophisticated tools like Venomics, there's still a vast amount of fundamental biological and chemical information locked away in the venoms of countless species that we haven't even begun to explore in detail.
It really feels like, despite centuries of fascination and decades of intense research, we've only just scratched at the surface of this incredible natural pharmacy.
That's absolutely right. The known world of animal toxins is vast and complex, and the unknown diversity is likely much, much larger. There's still so much potential for discovery out there.
This deep dive has been absolutely incredible, starting with that simple but absolutely crucial distinction between venom-needing injection and poison-needing absorption or ingestion. Really did set the stage for understanding these fascinating creatures.
It did. And from there, we saw just how incredibly diverse the world of toxic animals really is. It spans across almost all major branches of life, from tiny insects, spiders, and marine creatures like snails and jellyfish, up through fish, amphibians, reptiles, even birds and mammals, each group evolving its own unique strategies and chemistries for using toxins.
We really got into the nitty-gritty of the intricate biological machinery involved in delivering venom, didn't we? Especially focusing on the complex and highly evolved fang systems and muscular mechanics of snakes, seeing just how they achieve that rapid, precise injection. It's stunning engineering.
It truly is. And then we discovered the really surprising, almost paradoxical flip side, how these very same natural toxins, molecules designed by evolution primarily to harm or subdue, are proving to be invaluable resources for medical science. They're giving us critical research tools to understand life, new diagnostic tests, and even serving as the inspiration and direct template for life-saving drugs that treat major human diseases like hypertension, heart disease, blood clots, and chronic pain.
So understanding these distinctions, the biology, the chemistry, it not only clarifies a lot of common misconceptions people have, but it also reveals this deep biological complexity and, perhaps unexpectedly, the profound benefits we can derive from creatures we often perceive only through the lens of danger or fear. It's a potent reminder of the powerful, intricate chemistry happening all around us in the natural world.
It genuinely is. And it really leaves us with a significant thought to ponder, doesn't it? Consider this. The collected venoms and poisons of the world represent this massive, ancient chemical library, one that's been meticulously curated and refined by millions of years of evolution under intense selective pressure.
A library of potential discoveries.
Exactly. So given the proven potential we've discussed for discovering novel medicines, new research tools, and fundamental biological insights from these toxins, especially considering how many come from species that haven't even been properly studied yet. What crucial knowledge, what potential medical breakthroughs might we be unknowingly losing, perhaps forever, as biodiversity declines rapidly across our planet due to habitat loss and other human impacts?
It certainly makes you think very differently about the value of conservation, doesn't it? Beyond just saving species, we might be saving future cures. The danger these animals pose is undeniable, but perhaps so too is the profound potential they hold for our own future well-being.